Welcome to AP Biology. Today I want to talk about meiosis and how meiosis and sex shuffle variations, and so sexually reproducing populations have more variation than asexually reproducing populations. So first of all, why does that matter? Well, natural selection works on variation. It chooses the best variations for an environment. So where does that variation come from? Originally, it comes from mutation. So a mutation, when you copy your DNA, the copying of DNA happens before any type of cell division, meiosis or mitosis. So the mutations can actually get you new alleles. So that adds variation. That's the ultimate or original source of variation. And that happens both in sexual and asexual populations. But sexual populations have much more variation because meiosis and then sex can shuffle the variations. So mix up those alleles, and then natural selection can choose the best of those variations. So how does this happen? Well, there are three mechanisms that increase variation in sexual populations compared to asexual populations. Two of those happen in meiosis. They're crossing over and independent assortment. And then random fertilization comes when mom and dad love each other very much. So the crossing over, I think it's easiest to see with dad's chromosomes here. So dad has a chromosome that had originally come from his dad. So let's call that grandpa's. It had originally come from Gramps, and then one chromosome for this particular pair of chromosomes that had originally come from grandma. Okay, so what is that? What happens when he gives his DNA to you? So this sibling got dad's DNA, right? This is dad's DNA, so got half of this pair from dad, but most of it actually had come from grandpa, right? Papa maybe, and a little bit from grandma, right? But this sibling, actually, most of that chromosome pair, for most of that chromosome from this pair, well, the whole chromosome came from dad, right? But most of that DNA that they got from dad, most of it actually came from grandpa, just a little bit from grandma, right? So this is a whole reshuffling. It happens by crossing over and independent assortment. So that's what we're going to talk about next. So first of all, crossing over. Synapsis is when the chromosomes come very close. They line up almost gene by gene. They line up segment by segment, and then they trade or they swap pieces of DNA. So the chiasma is just that you see them kind of crossing there, but the synapsis is them coming over and matching up pair by pair. It's the non-sister chromatids that swap DNA, because if sister chromatids swap DNA, you'd get nothing different. So here's a homologous pair, and this one compared to this one, these are non-sister chromatids. So what you end up happening, this is maybe a little bit better. You'll see a tetrad just means you have 4 chromatids, right? So what you end up getting are sometimes these chromosomes called parental chromosomes. Well, right now they're chromatids, I guess. They look exactly like they had before. He went to work, honey. So those chromatids have no crossover. You can see the crossover right here, though, and right here, so 2 crossover. These are recombinant chromosomes. So recombinant chromatids right now, when they separate, they'll be recombinant chromosomes. They result from crossovers. And so right here, you get a chromosome that you're going to give to your kid, and it has maybe mostly DNA from your mom, but a little bit of DNA from your dad. Whereas this one, if your kid gets this, your kid is going to get DNA from you, but you had gotten that maybe all from your mom. So for those traits, they'll resemble more your mom. Okay, so how does this actually happen? There are protein complexes called cohesins that keep sister chromatids together. So they're represented right here. So these are the cohesins. You got a bunch of them here at the centromere and some right here. So there's an enzyme called separase. I don't know if you can see that. Separase. Separase is an enzyme that will cleave or will break the cohesins. So cohesins are cleaved or broken along the chromosome arms in anaphase 1. And so you might remember in meiosis 1, it's the homologous chromosomes that are separated, right, into 2 different cells. And so the cohesins hold the sister chromatids together, and you might think, well, no problem, because we're separating out homologous chromosomes. I don't need to worry about it. But you do actually need to worry about it because this chromatid, this piece of the chromatid, was originally on the other chromosome, right? And so if you don't separate out right here, then this one won't be able to join this one, or maybe the whole thing will get stuck together, or the chromosomes will break. Bad things will happen. So anyway, in meiosis 1, it's important to cleave the cohesins that hold the arms of the sister chromatids together. And then during meiosis 2, specifically anaphase, right before anaphase happens, right, you've got to separate out actually this part, the part at the centromere where the sister chromatids are still held together. And so same enzyme. So the enzyme is called separase, and it's breaking down these cohesins that are the molecules that hold the sister chromatids together. So just definition of recombinant chromosomes, it's going to combine DNA inherited from each parent. So this would be a recombinant chromosome that ends up here and here. And these would be the parental chromosomes. Crossing over happens pretty randomly. You might have it cross over a couple times, 3 times, or you might not have any crossover for a particular chromosome. Okay, so this just shows you some crossing over here. So I think the blue DNA can't remember where I stole this, but I think this person maybe has Irish DNA here, and I think the green one represented Jewish DNA. And so you can see chromosome number 1 pair. One of them inherited from one of her parents, didn't have any crossover at all, and this one just had one crossover right here. You can see a few crossovers. This means, I think for her, she had a grandparent who was Irish, a grandparent who was Jewish, another grandparent maybe who was Italian. I can't remember. But there wasn't a whole lot of crossover because those would have been crossovers from her parents. Whereas this guy is Latin, and look at all the crossovers. The pink represents African DNA, the yellow represents Asian DNA, the blue I think represents European DNA. Look at all the crossovers with him. That means it's not just his grandparents who had come from one area of the world. It went back many, many, many generations with him, where he has ancestors from a long, long time ago who came from all over the world, resulting in all these different tiny little sections of chromosomes, which is, I don't know, kind of cool. All right, so the next thing that adds variation is independent assortment. And so the chromosomes can line up, the chromosome pairs in meiosis 1, metaphase 1, right, of meiosis 1, metaphase 1. They can line up kind of however they feel like lining up, with the maternal on one side and the paternal on the other, or they could get flipped, right? It's very random. And so at anaphase 1, the homologous chromosomes are separated. And so let's say there's some organism that has a diploid number of only 4. They've only got 2 sets of chromosomes. They can line up this way, or they can line up this way with the maternal ones on one side or the maternal ones on different sides, right? And so what you would get is these combinations or these combinations, depending on how they ended up lining up in the beginning, right? Now imagine that instead of having 2 pairs of homologous chromosomes, imagine you have 23 pairs of homologous chromosomes and all the different ways those can all line up. And so for humans with an N of 23, a haploid number of 23 or 23 pairs, 2 to the 23 gives you 8 million, different combinations of chromosomes. That's without even considering crossovers. So we have lots of variation possible in our gametes. And then random fertilization, any one of dad's sperm might end up fertilizing any one of the eggs. Well, there's only going to be one egg usually released, but which egg is released is kind of random. All right, so that gives you, with all of these things, independent assortment, crossing over, and random fertilization gives you 70 trillion diploid combinations. Anyway, just think a lot. Okay, so it's this reshuffling of alleles that gives sexual populations much more variation than asexual populations, even though mutation happens in both sexual and asexual populations. Why does it matter? Well, evolution works on variation. And so if the environment is changing, the populations with more variation are more likely to survive. So organisms with the best variations for a particular environment will survive and reproduce. Yay.